So my presentation is based on the paper I have published back in 2018, which is one of the key outcome of my PhD project. And the reason why I selected this paper is that, and the species is considered as highly invasive species for many countries, and it alarms ourselves the presence of potential invasive species in our country waters as well. And of course, other reason is I am very fond of biofouling, and I hope after this presentation, you will find them as interesting just as I did. So starting my presentation, this is the outline of my presentation. If I give you an introduction, we know that long distance of travel has increased very highly as a result of globalization of world trade. And here, shipping plays a major role, transporting more than 90% of the commodities. This may facilitate the global dispersion of organisms to where they have never existed, and later they become invasive to the location of introduced, especially in the environment conditions which are very much favorable for them. Being an island country, the pearl of the Indian Ocean, located in the east-west marine route with a considerable amount of ship traffic, which is ultimately lead to the risk of introduction of exotic species into our country water. So this is the very old picture of Colombo Harbor, which was taken back in 1937. So when considering species introduction, as you may aware, there are two different pathways for species introduction. First one is intentional, and the other one is unintentional. Presently, the intentional introduction considered as the major cause for increased species introduction around the globe, where ballast water and hull fouling considered as major vectors. And recently, it was found that more than ballast water, biofouling considered as the major vector for the dispersion of geographically distinct species around the globe. And so, hull fouling accounts for more than 50% of species introduction around the world. However, the saddest thing is the studies on biofouling organism is very poor for many parts of the world, including Sri Lanka. And when considering maritime traffic and species introductions, the maritime traffic has been identified as a primary pathway for marine non-indigenous species introduction into around the globe, which ultimately lead into homogenization of species. With the development of new technologies, the volume of shipping and the diversity of vessels have increased, and together with the reduced transit times, the probability of species being transported to remote areas have also increased, especially in recent years. So before we move further, there are some terminologies that I want you to know. So first, let's see the difference between alien species and invasive alien species. When here we call alien species when they are introduced outside the natural geographic range as a result of anthropogenic activities. And these invasive species and these introduced species become invasive when they start competing against or preying on native species, which can lead to the extinction of native species. And this invasive species recognized as one of the top 5 threats of losing of biodiversity. So simply, when the alien species modified ecosystems, habitat, or species in the location of introduced, and when they start to cause severe effects on the ecology, economy, and health, we call them as invasive species. So keep in mind, not all alien species become invasive. In this slide, you can see how introduced or non-indigenous species are introduced from ship hull become invasive in the new environment. So when the adult fouling organism release their dispersal stages to water, they have to find suitable surface to complete their life history. The story begins when they find a ship hull as a suitable surface. So what happened next is they colonize in there and transport to other places, to new places. And next, they contract new structures in there or release their propagules to water. Then they stabilize in there and spread into nearby areas, altering native ecology, economy, or health. So when these species start to affect on ecology and economy, we call them as invasive species. So now let's see what biofouling is. So if I break down the word biofouling, bio means living materials. Fouling means attachment. So simply explaining, biofouling is the attachment of living materials, and this could be both animals or plants. And if I give you the scientific explanation for the biofouling, it is the accretion of micro and macroorganisms onto a surface which may have exposed to environment over a long period of time. And marine biofouling, we call it the settlement, attachment and growth of marine plants and animals onto a natural or artificial surface which are completely or incompletely immersed in marine water. And these artificial surfaces can be dead animal surfaces or even artificial structures developed by humans. And this biofouling is a process and it consists of four main stages. First, the formation of conditioning film, and the secondly, transportation of microbial cells into a conditional film that was formed over the surface, and then the immobilization of bacteria on the surface, and thirdly, the development of microbial biofilm, and finally, the settlement and growth of macrofauna. And when considering the fouling community, and it consists of two different branches. First is the microfouling, and it consists of protozoans, cyanobacteria, and bacterians. And second layer is the macrofouling. And macrofollowers are two types, as hard followers and soft followers. And hard followers include calcareous species such as barnacles, tube worms, mussels. And soft macrofouling includes species like ascidians and soft tube worms. And here in this photograph, you can see some of the specimens I have collected during my study. So the research gap is where even though we think biofouling as a, we have recognized biofouling as a major mechanism of introducing marine organisms. Concern on biofouling is very poor for Sri Lanka due to lack of interest and difficulty in taxonomy. Therefore, we have recognized that understanding of the structure and dynamic of the existing fouling assemblage is very much important. And also, it is very much important to know what is baseline to understand, to recognize the introduced and invasive species among the assemblage. So here I want to point out the only detailed study conducted on biofouling organisms was dating back to 120 years ago during colonial period. So here in this paper, I have discussed about bryozoans. So before I move further into my paper, I would like to introduce who are bryozoans is. So bryozoans are colonial invertebrate present in both fresh and marine water, but majority of them are inhabited in marine water. And they are considered as a major component of the fouling community, and their history goes back to early Ordovician period. And these bryozoans are moss animals. We commonly call them as moss animals, and they are almost and exclusively colonial. They are also suspension feeders, and their colonies grow on rocky substrate, but many other solid surfaces are also used, and their colonies range in appearances from gelatinous blobs to a bushy or tree-like form, which are called as erect bryozoans. Here, the species that I'm talking today is an encrusting bryozoan, which is also a calcareous one. So these colonies comprise of series of interconnected and genetically identical individuals. We call them as zooid. Individual zooids are microscopic, but these colonies can be relatively large. And here in this photograph, you can see a plate photograph of Schizoporella errata, another potential invasive species I found from my study. And their colony is quite huge, but their individuals are microscopic. And there are different types of individual coexist in this colony, and they are specialized for a particular function. And here I have listed some of those zooid types which are specialized for certain activities within the colony. And this feature considered as one of the key factors in explaining the success of this bryozoan within the fouling community. And here you can see a detailed image of a zooid. So there are certain features of these bryozoans that make them very successful organisms in the fouling community. And these many species are vitally tolerant to different salinity ranges, and they can settle on different range of substrate, and they have a very high reproductive potential, and also they are very fast growing organisms. So due to all these characteristics, bryozoans ranked as one of the most common fouling organisms in the coastal marine environment around the world, accounting for more than 6,000 species. So in this slide, you can see interesting bryozoan species I found from my study, and I have circled the species that were found as potentially species found as alien, and this is the species that I am talking today, Watercypora subtocata. And for your further information, I have included here two other erect forms that were found as non-indigenous to Sri Lankan waters that were found from my study. Moving to the methodology, I have selected Colombo port as my study site for my project, and apart from that, I have conducted my work in adjacent coastal waters to see the spread of the non-indigenous species I have found from my study. And these locations I have selected for my study were based on location-specific conditions and the guideline provided by the Center for Research on Introduced Marine Pests, where they are conducting work on biological baseline surveys in port environments. So these are the locations, 8 locations. So this is the structural view of my settlement panel, and these samples were collected by using these settlement panels, and there are this was fabricated locally, and there are horizontal PVC pipes and aluminium plates, and I have submerged 4 different depths. These panels were submerged in 4 different depths with 1 meter increment, where the first level was 1 meter below the surface, other one at 2, 3, and 4 meters. And I have deployed 2 collectors. One was permanently located throughout the study for 4 years, and the other one was used for monthly replacement to study the seasonal settlement of organisms. And here you can see an actual view of the settlement panel I have used for sample collection. So this paper was based on the initial study I have done back in 2014, and afterward I had to modify the collector because my previous collector destroyed with the environment conditions. So that's what research are. We have to modify our methods. So I have modified the previous collector into this, and I kept a set of plate as a backup because many of my structures got damaged. And here you can see the actual photograph of my collector deployed in passenger jetty in Colombo port. So the samples were collected from these panels by pulling the entire collector out of water, and We replaced one vertically and one horizontally oriented plate at each depth. And the samples were then chilled and transported to the laboratory for both qualitative and quantitative analysis. And I have preserved specimens for further references in the University of Museum of University of Sri Jagannathapura and in the marine laboratory. For the long-term panels to study the succession of falling community, I have deployed these structures throughout the study, and I have took still photograph of each plate for the both side, both inside and outside of the plate. Here also I had to take the entire collector out of water, and the samples were analyzed by using the photocop software. The species identification was done in two steps. First, I did the rough identification using available databases, identification guides, and those stuff. And then I went for taxonomic confirmation whether the species I have identified previously is completely right or not. So here I have used scanning electron microscope image projection systems, simply known as MIPS. And here I have considered 5 morphological features given in detailed research publication. And then I have determined their status, whether they are native or introduced, using databases and research publication, as well as using personal communications. And here are the morphometric measurements I have taken to confirm some of their characteristics with available identification guides, especially research publications. Here I have considered width and length of zooid, the individual of the colony, and the length and the width of the orifice, the opening, and the sinus, length and width of the sinus. So moving to the results and discussion, when considering the relative percentage of richness of major species group found in Colombo port, Bryozoans account for 8% of species richness. And here are the colony photograph of 10 species I have recorded at the end of my study. And among these, 8 species were encrusting, and remaining were branch forming erect bryozoans, that is Bivigula neritina and Bivigula simplex. So considering the species described in this paper, X Vorticipora subtorquata, it is an encrusting bryozoan, and it was recorded only in 2 of the sampling locations out of 8, and it had a very low covering percentage compared to other encrusting bryozoans I have recorded from the same location. And the most significant finding of this species is it is the first record for Sri Lanka to Colombo as well as to Sri Lanka. However, with their relatively low covering percentage and their presence in replacing settlement panels or the short-term community analysis, it confirmed that species is relatively recently introduced to a port environment. And here you can see the scanning electron photograph of this species, which I have used to analyze their fine morphological features. So this is the general classification of Vorticipora. It's belong to phylum Bryozoa, class Gymnolimata, order Celiostomatida, and family Vorticiporidae. And there are certain distinct features that used to identify this species from other bryozoan species I have collected. And as I mentioned earlier, identification of encrusting biofouling, encrusting bryozoans follows a significant features of the colony, especially their detailed features of zooid and their operculum, their sinus features, presence of pores, and likewise. However, considering Vorticipora, it was quite difficult to identify this species from rest of the Vorticiporidae because they lack many of the characteristic features present in other bryozoan species like Avicularia. I have mentioned earlier Avicularia, and the orifice likewise. However, when the identification was based on their operculum and presence of two distinct uh, uh, dots over here. That was the main thing, and their morphometric measurements that I have used to identify the species. So considering its colony, the colony is dark or black with bright orange outer growing edge, and the colony is unilaminar, that means only one layer, or it can be multilaminar depending on their growth, and it consisted of comparatively large, distinct zooids with these numerous spores. When considering zooid, as I said to you earlier, they consist of a very large amount of pores, and its operculum has distinct mushroom-shaped outline with a broad upper circular area and a rounded extension at the bottom. And this operculum is hugely pigmented with a broad dark central band with paler peripheral regions and two distinct white spots. It is much more clear in a previous photograph I have showed that include morphometric measurements. So, here, these are the distinct spots, shining spots. So, these are the length and width measurements I have taken to identify this species. And these measurements were found to be quite similar to measurements given by Ryland in 2009. And it was not similar to the species described as Watersephora subovoidea, which is very much similar to Watersephora subtorquata. And the Watersephora subovoidea was previously recorded back in 19th in Sri Lanka, but not Watersephora subtorquata. So when considering the habitat characteristics of this species, it's mainly inhabited in lower intertidal and shallow subtidal areas. They live in salinities ranging between 25 to 49 ppms, and their temperatures ranging from 12 to 28 centigrades, and they can live depths to the tens of meters, and they can settle on different substrate from natural to artificial and all kind of plates, but they prefer very much shady conditions. And this species also recognized as an early successional species, efficiently colonized on artificial structures in other countries. And it also found in my study because they were only present in my seasonal settling plates. When considering its geographical distribution, it is considered as a cryptogenic species for many parts of the world, and it's so their native region is uncertain, and as shown in the map, their introduced region include United States, Australia, New Zealand, and United Kingdom. So, what makes this species very successful invasive species in many parts of the world? So, because of they have wide salinity tolerance, and they are very fast-growing species, and they have a high reproductive potential, and they settle on wide range of substrata, and they are very efficient at colonizing artificial structures. But you might consider why this species was recorded as in a low amount of percentage cover. That's because of another species. I told you it before, that's Schizoporella errata, which I was found as that has invasive potential, and it has a competition between Watersephora subtorquata, and I think based on my studies and literature review, the act of invasive behavior of Watersephora subtorquata was surpassed by the presence of Schizoporella errata. At the end of my presentation, I will show you how this Schizoporella errata have become potential invasive in our port environment. So, considering the general impacts of these species, they form incrustations on man-made structures in oceans, and they impact society because it damage uh, instruments and structures that has aesthetic value, and likewise. And also they compete for space with natives and leading to displacement of native species. And also they provide secondary substrate for other invasive species, creating a non-toxic surface uh, to settle other species. However, as I said earlier, these species were recorded only in 2 of the sampling locations in Colombo port with a low covering percentage. So, therefore, I have recognized the monitoring is very much important to determine the magnitude of distribution in the Colombo port and also in adjacent coastal waters. So we can see whether these species have distributed further uh, with the as a secondary introduction, and we have to implement necessary management plans to prevent and shrinking and controlling the invasion to other part of the country and becoming invasive in areas where their this colonization is not uh, not surpassed by other species. So when considering invasive species or potential invasive species, we have to talk about managing how to manage these species. So when it's come to risk assessment and management, we say that an ounce of prevention is worth a pound of cure. So therefore, the prevention should be taken by using these actions. Here we have to say no to intentional introduction for those. For this, we have now implemented act to say no to international introduction through signing to clean the hulls before entering into our waters. And here also unintentional introduction should also be minimized, because from one of my studies I have found that marine debris or marine plastics also act as vectors for introduction of this kind of potential invasive species. And also we have to conduct more baseline surveys, surveillance, monitoring and contingency planning. And also community participation and awareness is also a must. So these are the references I have used for the presentation, and thank you. So before I say goodbye, I told you that I want to highlight one of the species I have recognized as alien, and it was at the end of my study, I have found that species have a potential to cause, uh, potential to become invasive in our port waters as well as in our country waters. So in this graph, you can see how the bryozoans have progressed over the throughout the period of my study in the long-term test panels I have deployed throughout this period of my study. And here you can see how they have increased over the years. And this graph, you can see how this particular species, Schizoparella errata, have progressed over the community, and how it have uh, uh, covered whole plate area that I have used to collect the community biofouling community. In this photograph, you can see that how the species have covered the whole plate area. In this slide, you can see how the species have gradually developed on the surface. You can see its progress. This is another potential invasive species, and this Schizoparella errata even cover that species as well. And I could also found there are interspecies competition, especially for space. And in the left, you can see how they grow over indigenous bryozoan species. And these two species are indigenous to Sri Lanka. And that is Electra bengalensis or Arbopecula bengalensis. And in the right side, you can see how it grow over another bryozoan species called Hyporhina indica. And here you can see how they grow over tunicates. This is after a month. You can see how they can even grow over soft biofouling animals. This is a colonial tunicate. You can see how they grow over these animals, and some of you might know that tunicates also have natural antifouling properties, and even that can be inhibited by the growth of this potential invasive species. Here you can see how they grow over other soft falling species, tube worms. And in this photograph, you can see how they provide substrate for other falling organisms. And the problem arises when these species start to provide substrate for other potential invasive species. And another important point is they can settle in multiple stages of the succession, even in the early stages as well as at the later stage of succession, because muscles settle at the end of at the later stage of the succession. So another thing is that I want to point out is what happened when these species become, this potential invasive species become a climax community. They see this colony or the community break out the whole community, and they have the succession have to start back from the beginning. So this is what happens when an invasive species or potential invasive species take over a native community. They just simply alter the whole community, acting as both allogenic and autogenic engineering species. Thank you.
Thank you very much, Kalpani, for that, Dr. Kalpani, for that very interesting and novel research area. So now it's the paper and the session is open for discussion. So if you have any questions, please raise them now. Kalpani, I have a question. I think it's really interesting, the fact that because this is a novel research area in our country, and you have developed an important device to study the impact of biofouling in ports. So my question is, like, in your research slide, you have showed like the species, the richness of species. Like there are a number of species that you have found using this device. So have you tried considering their like invasiveness to assess the invasiveness of the other species except this bryozoa? Because using this device, like we can do so many studies to see the invasiveness of all these species.
Yes, Dr. Maheshi. Actually, the device was developed to study the whole community succession, and to determine the invasiveness, we have to assess both the impact on the ecology, economy, as well as to health. Here, I have studied only the ecology of species. Actually, the main study was to determine the community composition and their interactions. And what the key outcome is that we can determine the species which have potential invasive activities. So that is what the outcome of this project. But as you said, we can keep these devices for a long period of time, and we can, assess, we can conduct a monitoring program how this species grow over a long period of time. The other species, I have found many other species that have potential that are non-indigenous, but among these, it's the Schizoborella errata, and there is another species, Eulex tulipa. I have showed that that species was also covered by Schizoborella errata, and likewise there are many other species that I have already identified through this device. Through the community analysis, I have identified many other species as well. So just like you said, yes, this device can be used as a monitoring device to further monitor this kind of species. And my structures are already um, in the Colombo port, but with the situation I couldn't make a visit. But the community is keep developing in the structures, and I think yes, we can definitely continue the study to determine the invasive capabilities in point of ecological point of view. Uh, but for the to determine whether these are invasive, we have to go for we have to determine the ecological, economical and health impact along with the ecological impacts. Yes.
Yes. Thank you, Dr. Kalpani, for that clear clarification. Are there any other questions?
Kalpani, thank you very much for the interesting presentation. Do we have any evidence for these species being, having any effect for the native fish diversity?
Yeah, it's actually, Dr. Sachini, it's indirectly affecting because, for an example, which I would like to talk about the species that I have already identified as a potential invasive species. This is because this species was controlled by that Schizoborella errata. The Vatocipora was controlled by the Schizoborella errata. And if I talk about the species that I have already recognized, I found that the species can alter the community composition, and it's completely destroy the development or the succession of the community in the natural environment, which can ultimately lead into the loss of biodiversity and the homogenization of the species in the environment that can lead ultimately lead to the to affect the fish and other species in the environment. Not directly affecting, but indirectly it affects because the
Okay.
community composition changes.
Okay, thank you.
Kalpini, yes, can you hear me?
Yes, madam.
Yes, Kalpini, I was just thinking that you said that one of the problems with this study was the taxonomy, that you found it difficult to identify certain species of bryozoans and other biofouling species, and that you used these microscopy methods, I think, with standard keys and also publications.
Yes.
Am I correct? Yes?
Yes, madam.
Yes. So, did you try to sort of identify them comparatively using specimens in our two maritime museums? We have two maritime museums in Colombo port and
No,
also in Gaul.
previous collection is not in the country. It's in Liverpool. So therefore I couldn't compare my specimen with them. But I have, I got regional specimen collection from the papers. Not I couldn't physically compare the specimen, but I went through the publications and identified the specimens using those features. Apart from that, I'm continuing molecular confirmation of these species, and some of these species need to modify certain extraction methods. So the work is still progressing, madam.
Yes, yes. Okay, thank you, Kalpani.
Kalpani madam, I saw that in the biofouling process, there was a initial step, the most like the first step In that diagram you showed, I can't remember the name, some priming or something.
Sorry?
There was initial step. What's the first step you mentioned?
Formation of conditioning film.
Yes. What is that conditional film is made of?
It consists of polysaccharides and debris and those stuff that facilitate the settlement of microbes.
Yes.
The surface is conditioned by using many organic and inorganic materials in the environment.
Okay, it's formed from the elements present in the marine water.
Yeah.
Okay. It doesn't involve any bacterial or any other
Later on, the conditioning film is attracting the microbes, and that's what makes the microbial biofilm. Actually, I didn't consider micro part in my study. I only considered the macrofouling part.
Okay, madam. So those anti-biofouling paints and coatings all are made to make it resistant to these
microbiofilm. Yeah.
Microbiofilm. Okay. Thank you, ma'am.
Hello, Kalpani. This is Preeti.
Yes,
thank you for that lovely presentation. I am just wondering how we are going to take this forward using our department as a base. What are the plans you have?
Yes, madam. I have already working on applying for a grant, and for the other thing is what I really like is that this subject can relate to anything in ocean. For an example, if we consider fishing and this biofouling fall on fishing gear and cage structures in aquaculture, and they impact the fishing practices. So that's just an example, and I can interact that into that my discipline is marine ecology and fisheries, and concerning marine ecology, I basically studied the ecology behind this biofouling community. Apart from identifying the species, so I have already applied a grant incorporating both fishing and the marine biofouling.
Okay, all the best then.
Just to initiate my research interest.
Good. Okay, all the best. Thank you.
So we can wind up today's session. And thank you for the audience for your interactive involvement of this journal club presentation. Thank you very much.